The first group of tissues we're going to be looking at today in lab are connective tissues. And we're going to be looking at 3 types of connective tissues. We're going to be looking at connective tissues proper. We're going to be looking at supportive connective tissues, and we're going to be looking at fluid connective tissues. So the first batch of connective tissues that we see are going to be connective tissues proper. We are going to be looking at 3 examples. And the first example is going to be areolar connective tissue. And this is the most generic of the connective tissue types. It has a very open framework, and it also has a lot of different facets or features that we find in connective tissues. So it's one of the most generic, and we'll see different things that we can see in other connective tissues as well. So for example, we can see collagen fibers, which are these sort of purpley stained fibers here, which I might be able to make a little more visible here. So not quite that low. Bring up the exposure a little bit, and you can see all of these purple things in the background, this meshwork of purple, sort of thicker purple fibers. Those are collagen fibers. Then we have these darker fibers. They show up either black or dark blue or sometimes dark purple, the little thin ones, and those are elastic fibers. And you can see them interspersed all throughout here. And then we see the nuclei of the fibroblasts. And the fibroblasts make these collagen fibers and these elastic fibers. Now, collagen fibers and elastic fibers are both protein. When we look at the background, that's the white stuff, and that's the ground substance. And then there's this thick molasses consistency stuff that has a lot of proteoglycans in it that's sticky. It's really good for keeping pathogens at bay. Right now, we started with this. I started the video with the low power objective, although of course I started with the scanning objective, I just didn't show it. And now I'm going to bump this up to the high power objective. Now to do that, I'm going to need to adjust my exposure on this particular microscope, and then we can start refocusing. And now we can see areolar connective tissue up close and personal. Here you can see a collagen fiber right here. Here you can see an elastic fiber. Here's another elastic fiber. These little stringy ones are elastic fibers. Sometimes they look kind of kinked, like here. Sometimes they're wavy, but usually in areolar connective tissues, you'll see a lot of straight ones. All these dark things are the nuclei of the fibroblasts, and then we have our collagen bundles in the back. Now, one thing to be aware of is that we have different stains that can be applied to this. Sometimes the collagen fibers are not that clear. Sometimes they stain a dark purple, sometimes they stain kind of pink. So what I'm going to try to do is find another example of the areolar connective tissue and see if I can find one that has a slightly different stain to it. Now here we have another example. In this case, I've started the video with the zoomed out view and the scanning lens. So now I'm going to flip it up to the low power objective, give a little refocus here. Try not to focus on the artifact on the slides there. And now you can see how we have a slightly different coloration in the stain here. You can still see the elastic fibers stained dark. The nuclei of the fibroblasts stain this dark blue, purple. And now here you can see some of the collagen fibers look more pink than they did in the other slide. Now I'm going to bump it up to our high power really quickly. And here it actually looks quite similar to the previous slide. Coloration may be a little bit different, not that much, but the point is sometimes you'll get slides where the collagen bundles are darker, more purple color. And here you see some collagen bundles back here, lots of nuclei, and here we have lots of elastic fibers too. And just to give you an idea of the depth of field, we can change the depth of field. We can bring some of our elastic fibers into view, into focus, and some of our nuclei come more into focus, and then we can change the depth of field again. And now some of our collagen fibers come more into view. So this is just an example, as we've seen before, about how you can change the depth of field. So that is our areolar connective tissue. Once again, we've got collagen fibers that are this sort of pinkish stuff. We've got the elastic fibers, which are these darker colored ones that can be almost straight, or they can be quite kinked or a little bit curvy sometimes. We also see the darker nuclei of the fibroblasts, and in the background we have the ground substance. And the ground substance, as we have said, is not conducive to pathogens swimming through it. So it's a good first defense of keeping pathogens from being able to get around in here. So it's a good way to trap pathogens. And as we study connective tissues, we see we have some resident macrophages, which are cells that live within connective tissues that will look for pathogens and try to destroy them before they can invade the organism. Next up, we have adipose tissue. And what you'll notice about this immediately is it looks kind of like bubble wrap. Sometimes it's clear, sometimes it stains this pink color. And what we'll see is we see these large open spaces. Now this, of course, is the scanning view, so we're kind of zoomed out. And what we'll notice when we zoom in on it is we'll see that these cells, well, they're adipocytes. They're very closely packed together. And in most connective tissues, you'll see that the cells are sort of sparsely dispersed throughout the tissue. And you have all these other things within the tissue, like the matrix, like the collagen fibers and the protein. In this case, these cells are packed together. So I'm going to go ahead and move it up to the the low power objective now, and we'll refocus it so that we can sort of see a little bit more about the structure of these cells. Now, these big open spaces in the center are what we call the fat vacuole. This is a specialized part of the cell that is going to be what stores fat. So it stores fat in the form of a triglyceride. So this thing is just full of triglycerides. And all the cytoplasm of the cell is smashed out to the side. So the very edge of the cell is the cell membrane, which is really hard to distinguish from the cytoplasm, which is this pale pink stuff, because it's kind of smashed out to the sides of the cell, smashed up against the cell membrane, because this fat vacuole is taking up the entire or most of the space in the cell. And then we have the cell nucleus, which is also smashed out to the outside of the cell, and it has a sort of signet ring appearance, like a gemstone in a ring. So if we move up to the high power objective now, we will see where you can see the fat vacuole, the center part, the nuclei, which are staining here almost purple, and all this stuff on the outside sort of smashed up to the cell membrane is the cytoplasm. And the cytoplasm really does not take up much space in the cell, because most of the space in the cell is taken up by the fat vacuole. So each one of these individual cells is an adipocyte, and each one has a nucleus and a fat vacuole, and all the cytoplasm is smooshed out to the side. Now, a quick note. In some slides, this is almost clear, but nevertheless, it's going to have the same morphology, where you have these big blobs in the middle that look clear, and this is where we're going to have the fat stored in the fat vacuole. In some of the other slide preparations we might see, this part here might also be clear, but you can still see a faint shadow or a gray part here where the cytoplasm is. And the cell nucleus also is fairly visible in those slides. But this is a very good example of adipose tissue right here. The first two types of connective tissues proper that we looked at were areolar connective tissue and adipose tissue. Both of these are considered loose connective tissues. Now we're going to look at a dense connective tissue. And in this case, we're going to look at dense fibrous regular or dense regular fibrous connective tissue, in which what we see are densely packed fibers, collagen fibers. In this case, this is going to come from a human tendon. And remember, a tendon attaches a muscle to a bone. And so we have these collagen fibers that are arranged in bundles like ropes, and this gives it a high tensile strength so that it can resist forces in one direction very nicely. And we're going to look at the regular connective tissue, which means these rope-like bundles, as opposed to the dense irregular connective tissue, which has a very similar construction in terms of the fibers, these collagen bundles, but they're going to be more woven together like a meshwork, so more like a meshwork rather than a rope. In this case, we're going to look at the regular connective tissue, which is more rope-like. And where we get this tissue from is a tendon. So let me move this up to the low power objective very quickly here and refocus. And one of the things that you might see on some of these slides, here we see some very nice example of some dense fibrous connective tissue. And you can see these collagen bundles, or these pink things here, And the darker staining, these darker staining parts, these nuclei, are nuclei of the fibroblasts that are going to maintain these or make these collagen bundles here. So the collagen bundles are these long fibers. And then we have the nuclei of the fibroblasts that make the stuff, and they're usually kind of elongated. And they tend to follow, sometimes they can be wavy and almost angular. One of the things to be very aware of, as I mentioned in one of the previous videos, is these nuclei can stain anywhere from pink to dark purple. And oftentimes on first blush, it looks a lot like smooth muscle because of the coloration and the fact that you have this background of pink with these nuclei interspersed between them. But if you look very carefully, it's the nuclei of the fibroblasts are in between the collagen bundles, whereas if you look in smooth muscle, the nuclei are inside the cells. The other main difference is the pink part is actual collagen fiber. So this is protein fiber, it's not a cell. Whereas in smooth muscle, the cells, if you look at them, you can see they have this tapered appearance when we look at them longitudinally, and you can see that they are individual cells, where here we have these bundles of collagen, these pink bundles of collagen, and interspersed between the pink bundles of collagen we have these nuclei of the fibroblasts. These can stain anywhere from light pink to dark purple. I'm going to zoom in on it a little bit and go to our high power here, and then we're going to change the exposure setting a little bit so you can see it, and then I'm going to zoom in on it or focus in on it some more. And now we can see that the structure is a little bit different from what we saw with smooth muscle, because you can see the pink parts are obviously fibers, they're not cells, and the darker staining nuclei are sandwiched in between these fiber bundles. So now what I'm going to do is I'm going to move the slide around a little bit, and you can see some of the nuclei here. This one's sort of right on the edge here. Here's one. Here's a nucleus. And then these are our collagen bundles. So we can look around the slide, and one of the things that I'm going to try and show you, which you will find in some of these slides here, we've got the collagen still. We've got our collagen fibers. We may have to refocus the slide a little bit as we move through it. We can see the collagen bundles, and we can see each of the nuclei of the fibroblasts in here. But sometimes, very occasionally, on these slides, since these are tendons and they do connect muscle to bone, sometimes you can find examples of human skeletal muscle in these slides. Now, on this particular slide, I'm not seeing any. But one of the things that you should be aware of is that in some instances, you might find muscle tissue on the same slide as dense fibrous regular connective tissue.